Hello everyone. In this video, we will be discussing what was the environment and what did they eat, referring to peoples and populations of the past. This is in part a review of Renfro and Vaughan's 2015 Chapter 6 Archaeology Essentials chapter, but I've expanded it with different areas of interest and different sources. So let's start talking about what we can learn from archaeological evidence regarding people's environments and their diets. So we're looking at reconstructing past environments and understanding what people ate in terms of their meals individually on short-term occasions or their diet over long periods of time. These questions are really the most fundamental or basic questions that the archaeologist can ask in terms of reconstructing environments, environmental reconstructions or past environments. They can help us answer questions about general observations through time, referring specifically to global climate change. So when we investigate environments, typically it's on a hemispherical level or sometimes on a global scale. But we also can learn very specific information about environments and climates on a localized or even a regional scale. So tree rings are a good source of annual records. They record primarily precipitation levels relative to one another. But the nice thing is we know that certain kinds of trees will leave a ring every year, so it can provide a wealth of information and serves as really an accurate benchmark for chronological controls and environmental controls. And then we have ocean cores and ice cores, which operate fundamentally on the same basis as or concept of tree rings, where sediments are deposited at a regular interval. For example, in ice cores, it snows every winter quite a lot on Greenland. And because it's so cold there year round, that snow accumulates year after year after year and becomes stratified in the ice sheets. So before they melt, scientists are rushing to core ice caps in the north on Greenland and down in Antarctica as well, to get this worldwide record of temperatures, atmospheric conditions, and a range of different information, precipitation. There's a lot of information that is stored within these ice cores. Likewise, ocean cores, material and plant life and organisms in the ocean, when they die, they settle to the bottom of the ocean where they become stratified as well. So by coring the floor of the ocean, we can learn quite a bit. These are some examples of ocean coring. As I mentioned, if we're looking on a global scale, it's good to compare global climate changes with regional, what we would call proxy records. Tree rings being really good for establishing climate and precipitation information on an annual basis, so highly accurate over thousands of years, and in some regions going back as far as 10,000 years. Ocean cores offer much broader patterns, but over much more elongated periods of time. So if we're looking at sea and ice cap cores, the stratified layers in the ice or on the ocean floors pile up, and through the law of superposition and the concept of stratigraphy, we know that those layers that are lower in that column are older. And we can also date some of the layers directly. In some cases, within the last 50,000 years, we might be able to carbon date items that are trapped in those stratified layers. But there are other ways of dating further back in time. One in particular species that is important, particularly for ocean floor coring, is foraminifera. Foraminifera are small organisms that live in the ocean. They're very sensitive to the ocean temperature, and they preserve in their shells the climate through what we would call a proxy record. So foraminifera are an excellent library of proxy records for environmental reconstructions. Different species are sensitive to different types of temperature changes, and they're essentially saving a particular isotope, oxygen 18 and oxygen 16. The ratio of these two isotopes can give us an indication fairly accurately of the ocean temperature. So these are regional correlations that establish global trends through time. And here in the chart, you can see a simple warmer and cooler wiggle line from different cores, and they correlate with each other, forming a broader pattern for all of the Atlantic Ocean. The Atlantic Ocean is a really important source of driving worldwide weather conditions and climate. And this one goes back 135,000 years. So we're interested in this because of global climate change. It's an important process that we're experiencing now for detrimental effects on human and animal societies as we continue to cause atmospheric pollution with greenhouse gases. We're driving up the average annual temperatures of our oceans and of our atmosphere, which is resulting in global climate change. Isotopes save the temperatures when ice was formed and piles up in these ice sheets. So we can go back in time and compare the natural fluctuations, establish global cycles. We know that the Earth goes through ice ages and glacial periods and interglacial periods where it's cold and periods where it's warmer, like the current present geological epoch that we are in. The next ice age we could expect sometime within the next 15,000 years if we were operating on a natural cycle. However, it's possible that human-induced climate change or global warming through greenhouse gas emissions is holding off or could stall the next ice age. Not that we would do very well with continental glaciers coming down out of the North Pole again, but on the other hand, we don't want the planet to be too hot or it could become completely uninhabitable. So in the current era, humans are inducing climate change. Of that, we have no doubt. Greenhouse gases are the highest in 440,000 years. So we have had periods where it was warmer and climate was radically more different. The issue is the rate at which we're warming the planet, and it's contributing to a sixth mass extinction event of plants and animal species on Earth, among other issues like environmental degradation, overhunting, and overexploitation. So this is what it means to be living in an era of anthropogenic or human-caused climate change. Greenhouse gases have increased at least 100 times faster in the recent century or 2 centuries than they have at any other time that we have been able to measure, going back hundreds of thousands of years. It's unnatural and it's completely human caused. Therefore, the implications are really unfounded, right, for this rate or speed at which we're warming up the planet. In geological time, this is a much more gradual process or shift. And biological species, some may go extinct, species change, that is a natural cycle. But in many cases, they're able to adapt if it's a more gradual change. So think for bison, for example, which is a Pleistocene, sort of large Pleistocene game that survived the last transition from glacial to interglacial cycles. Past carbon dioxide fluctuations that can be measured. So in a typical glacial period or ice age, we would have about 200 parts per million of carbon dioxide in the atmosphere. During a typical interglacial period, we'd have about 280. So you're really looking at a slight difference of less than 100 parts per million in the atmosphere that could result in a shift from a glacial period to an interglacial period. Now, today we're at 409 parts per million, almost basically 410 parts per million. This is dangerously high, and it's increasing. What are some indicators of warming climates past and present? If we can measure these through proxy records, we can reconstruct the climate of the past, and we can observe them in the present as indicators that we are warming the planet. We could look at average annual air temperature near the surface or higher up in the surface, like what is the average over a year period of our atmospheric air temperature. Levels of humidity go up when we warm the planet. Temperatures over the oceans increase when we put more greenhouse gases in the atmosphere. Sea surface then warms up as well. Sea level temperatures go up. The ocean heat content goes up overall. Sensitive fish and organisms can't handle that. Salmon, for example, are very sensitive to water changes. The level of sea ice will decrease. Glaciers in the mountains and snow cover will decrease. This is a source of fresh water for many communities that have urban centers around mountainous areas like Denver, Colorado, for example. Major cities in India that rely on Himalayan ice pack. Villages, towns, and urban centers that get water from Kilimanjaro in Africa, all in danger of losing snowpack, which is a form of storing drinkable water for not only people, but animals as well, rivers and lakes and streams. Those numbers will go down. So we'll see a reduction of snow packs in the mountains and of sea ice. Here's some data by NASA that shows the percent of carbon parts per million of carbon dioxide in the atmosphere going back 440,000 years ago, and this is before today, basically. Years before today, or this indication is 0 equals 1950, basically, but the trend, the trend is indistinguishable really from 1950 to current. We are in a straight line upwards. So you can see how it's fluctuated, and it's never really gone above 300 parts per million before, and we're off the charts here at 410 parts per million. So this is the reason why we know for certain that this is a not really a natural cycle, in that we're continuing to climb, and that we know the source of the emissions is from human activity. What can we learn from some of these different proxy records about past climates? Because people have been adapting to climate change for millennia. Well, we know that the Earth, through uniformitarianism, really does operate on principles that remain the same through time. So equatorial and polar temperature differences generate weather patterns and ocean currents. So oceans act as conveyor belts, moving energy around the planet and driving our local weather conditions, and over a period of time, our climatic conditions. So even levels of storminess and wind can be used to compare regional temperature patterns, and by analyzing the differences in through time of, say, equatorial temperatures and polar temperatures, we can get a sense of regional shifts and help reconstruct what we know about past wind patterns, levels of storminess, and other factors that affect continental scale patterns. So some key concepts that we've kind of talked about already: evidence from sea and ice cores are important. Ocean sediments and stratification of those, foraminifera, are climate proxy records. They save the isotopes like oxygen-16 and oxygen-18, and the ratio of those isotopes that are preserved through time can tell us about the average temperatures of the oceans for ocean floors. Similar types of studies for ice sheets and ice cores, which are stratified annually through the precipitation rates. So these give us climate proxies to study and reconstruct different regions, and it helps us construct a worldwide view of climate change through time. Evidence from tree rings are finer grained in scale, annual constructions. They're laid out every year. They tell us annual precipitation, and they're excellent chronological markers used as a calibration tool for radiocarbon method that would allow us to go back further in time. Really the longest dendrochronological record or tree ring record goes back about 10,000 years. If we want to reconstruct the vegetation environment or the plant environment, we can answer that question. What was the vegetation like in the past? And we know that plant vegetation changes through time depending on levels of precipitation, temperatures, climatic conditions. They can shift. These provide clues to the different animals and ultimately humans on the landscape. Shifts in plants by altitude also is an indicator of climate change. As the climate warms, we see plants going further and further up slope in elevation when they typically have a narrow range of tolerance. We see that shifting upwards until they, until the conditions surpass the peak, and then some of these species may go extinct in different regions. So we can analyze pollen from different places. One excellent source of stratified pollen through time are ponds and bog cores. Particular characteristics lead to better preserved sedimentation rates. You want it to be a place where pollen can rain down on the pond or the bog and then settle to the bottom and be preserved in the stratified layers of a bog or pond. So in pollen analysis, we would call this palynology. Pollen rain occurs mostly during the spring and summer, so it tends to be seasonal, and it preserves a distribution of pollen frequencies through time. And here you see a chart that measures the relative abundance of different species like pine or alder and birch, elm and oak, hazel, different types of nettles and grasses, and then a general activity on the right. So this is a typical example of a pollen chart. And this especially is from Northern Ireland, where it reveals forest clearing, the first appearance of farming, the dissipation of farming and the resurgence of forest cover, and then another cycle of clearing of the forest and the planting of grass crops. Pollen is best preserved in a rather acidic and poorly aerated environment, and that is really a peat bog is one of the best places for these kinds of conditions. So it's a level of acidity and low oxygen levels that lead to excellent preservation of many organic materials, pollen being one of those. Phytoliths are the really hard parts of plants, sometimes called plant opals in some archaeological literature. These are tiny particles primarily of silica. It's readily abundant and it's highly resilient. It preserves extremely well. Often you can find different phytoliths in hearths that were campfires, they were burned. Sometimes you can find them inside a pottery. If they were used for cooking, the phytoliths will preserve, and the cooking kind of helps preserve them in some cases. You see them mixed into plaster. Depending on what they're used for, you might find phytolith polish on stone tools that can be analyzed, a silica polish. This is especially true from grasses. Diatoms are another useful proxy or direct indicator of different species. These are single-celled algae. They're found primarily in lacustrine environments, often in ponds where the wave action is low. You might find them in brackish waters, but different kinds of diatoms and species live in different environments. So their silica cell walls are also resilient and they preserve quite well. They reflect levels in salinity, alkalinity, nutrient content, again, because they're so sensitive to changes. When you find them that different species are present, you can understand if the pond was fresh water, if it was brackish water, or if it was totally salt water, and different other conditions of the water through time. Other things are more obvious. You might just find the remains of plants. So here's some 4,000-year-old millet noodles that were still in the bowl. Somebody didn't do their dishes in northwest China 4,000 years ago and left their food in the bowl. It became stratified and buried, and you can see the impression of the soil that had filled the bowl, and in the bottom of that are these noodles. They preserve quite well. These are macro-botanical remains. Seeds and fruits often preserve well, especially if they're charred in a campfire. Sometimes you'll see the grains of different plant material, thinking mostly Neolithic seeds from grasses, barleys, wheats, rye, emmer wheat. Sometimes that is preserved in pottery if it's used to temper the ceramic, or clay bricks. We find them embedded in clay bricks where they were worked into the clay, or just impressions of them, which give us an indication of the different types of plants that were around. Seeds also preserve very well archaeologically, and we can find those. We can also extract environmental DNA from different soils depending on the preservation levels, and directly sample levels of DNA from different stratigraphic levels. Different kinds of plant residues can be found in cooking and charred, especially inside ceramics that were used to cook different plants. So pottery residue analysis is macro and microscopic analysis of different charred remains, can identify specific foods, plants in particular, that were cooked and eaten, presumably eaten. Wood remains are useful, often from campfires. If you sample the charcoal from hearths or remnant campfires, you can analyze these under a microscope and determine the genus and species of the woody plant that was burned there based on different cellular structures. This is useful for identifying surface vegetation. Here's an example of an acacia tree from Africa on a microscopic slide. So a few key concepts here that we've talked about with respect to reconstructing the plant environment. We can look at microbotanical remains such as pollen and phytoliths or diatoms, or we can look at macrobotanical remains such as seeds and fruits, plant residues, or the charcoal, wood charcoal. And then we can chart these over time, like in this example here, of different types of woods through time and pollen zones that match up very well. This gives us an excellent example of how to fully recreate the plant environment of a region, a pond, an archaeological site, or whatever through time. If we want to reconstruct the animal environment, we can again look at microfauna, or we can look at macrofauna. So microfauna, if they're very small, these tend to be better indicators of climate change, or more sensitive. So for example, the diatoms, more sensitive. That's a plant, but we can look at the biological aspect for the animal environment as well. These are better indicators of climate change. More sensitive to environmental changes. Bugs, beetles, insects tend to be more sensitive to temperature changes and other shifts in their environment. These tend to accumulate naturally also. Humans don't tend to go around and collect very small micro animals and introduce them into an archaeological site. So they're highly representative of the natural environment. They tend to accumulate naturally. Some have, as I mentioned, very specific climatic requirements. Now, macrofauna, you could find entire mammoths, like this example of Dan Fisher looking at the frozen baby mammoth that came out of the Siberian steppe. Very well preserved, even its stomach contents are preserved. These are often, though, not always deposited by humans or by animal activity, which can increase or decrease their level of preservation in the paleontological record or any archaeological settings. Humans will go out and hunt different species selectively, bring those parts and bones back to their archaeological site and introduce them there. So you see human intervention in the level that these are preserved across the landscape. So bone deposits may indicate human hunting selections, but you can also get natural die-off events where entire herds of mammoth are killed naturally and not involving human activity. So you have to determine if humans were involved or if they were not. Nonetheless, they are also not very good indicators of vegetation because many large animals can eat a wide variety of different plants. So you still need your plant reconstruction for paleoenvironmental records. They are an indication that the species was present at that time, and you can date them directly and determine like this is the last existence of Pleistocene horse on the landscape in this area, or this is the last known evidence by radiocarbon date of a fossil of woolly mammoth on a landscape. So it indicates the presence or absence, not necessarily the absence of a species. You may just not have that particular species preserved there. It is useful indicators of seasonal occupations depending on the type of animal. You may have migrating birds, you may have tooth eruption sequences in young individuals, you may have antler sequences from cervids where they drop their antlers at particular times of the year. This can offer clues to season of occupation of a site or use of that environment. Shifting now over to subsistence, or what people ate, how food resources were extracted from their environment by people. What types of foods were they eating? How did they feed themselves? What is the difference between a meal and a diet? So subsistence and diet, this different information from the archaeological record include, you may find the actual bones in an archaeological site. You can conduct a zooarchaeological analysis of the fauna from an archaeological deposit and say this is how many examples of this particular species was found at this site, and that changes over time. Sometimes there are written records or pictures indicating counts of animals, how many goats were traded for how much olive oil, and so on. And further back in prehistory, you can go all the way to the Paleolithic and look at Paleolithic rock art, such as that at the famous site of Lascaux in France, to tell us what was on the landscape and what were people most interested enough to create it on a mural of rock art, for example. Ethnoarchaeology may be able to help if people have ancestors or ancestral stories. They can talk about the changes and fluctuations of the different animals that they hunted and where. Zooarchaeology really is very informative. If you find the direct evidence of them in the archaeological site, you can count them and measure them and document them. Paleoethnobotany is a study of past human plant use. That's useful. So here's some Paleolithic rock art, late Ice Age, highly artistic. You see anatomically different animals that are represented there, in this case the woolly rhinoceros in Europe. Excellent indication of Ice Age fauna. Here is a grasshopper from, they're not very common, insects aren't common in rock art, but here's an example of a Paleolithic grasshopper depicted from the Magdalenian period of France. Insects are more sensitive to climate change than larger fauna. For subsistence and diet, we can look at food remains. Sometimes it can be problematic. The most direct way is to find it in human feces to interpret what they actually ate. So that is incontrovertible proof of consumption if you find it in the feces because it came out in the digestive system or in fecal matter. Butchering and burning and cut marks are all indications of food processing and cooking. It's a fair assumption that strongly suggests consumption. Why would they cook and butcher an animal if they're not eating it? But nonetheless, we do find a lot of evidence of these kinds of activities. So you can actually learn quite a lot from feces. There are studies at Paisley Cave that tell us different attributes of what is represented in feces by all kinds of different analyses. You can also tell the health of the individual, and here is a useful Bristol stool chart that analyzes different types and varieties of human feces as representative of the health of the organism. So what plant foods tell us about diet is quite a lot. And we can look at plants through macrobotanical remains, what plants were at the site, how are plants processed and used. We can look at different plant residues on artifacts. Are there chemical traces of plants in the pots and on tools? We can look at wild or domestic species of plants and how those change through time. What is the relationship of people with plants in terms of the whole process of domestication and manipulation? We know that processing of cereal foods is very time consuming. It uses a lot of energy. Here's all the different things it takes to process and deal with, say, barley or wheat or rye. And it doesn't even talk about the baking of bread. What plant foods can tell us about diets, we can look at chemical residues in the plants and the remains of plants. We can look at their DNA. We can look for proteins and fatty lipids in DNA. We can look for plant impressions, commonly in fired clay, as we've talked about. But none of this is not really proof of its significance, maybe in the diet. We can analyze plant residues on artifacts, microwear analyses that demonstrate different cuts if we're looking at use wear from tools. Phytoliths may show different types of grasses. You can do potassium iodine tests, which turn blue if there are starch grains present on an artifact. Different fats can be identified from fatty acids and amino acids to identify different types of plants or animals that were consumed. It all tells us about this lengthy process of domestication of wild plant and animal species, frankly. These were human management of plants, cereals, agricultural produce, and this transition from the upper Paleolithic hunter and gatherer and forager, where the subsistence was obtained by ranging through a natural environment, collecting and consuming, to a food producing economy, or the farmer, a Neolithic type farmer who is manipulating the plant and animal to the extent that it changes the genetic, biological genetic genome of that species to benefit humans, and in some cases a mutual benefit to plants as well and humans. This is a lengthy process of interaction of people in a symbiotic relationship with plants and their environment. Looking at diet and seasonality and domestication from animal remains. Animal remains are often very well preserved in different contexts. They may be from human activities, or they may be from natural activities. If you look at the sex and age of an animal, it can be useful in telling different types of things of the behaviors and the circumstances that led to them being deposited, particularly in an archaeological site. Seasonality can be determined from tooth eruption sequences. If you have bird fauna, avian fauna, these are sometimes seasonal indicators. If it's a migratory waterfowl that you say find in particular site in cooking hearths, you know that they were there at a particular time, on the assumption that migrating birds have a similar pattern today as they did in the prehistoric past. Analyzing bone taphonomy is very useful, like looking at carnivore marks, tooth marks, consumption marks from natural events in animals versus V-shaped cut marks from stone tools. You can tell the difference between chop marks and slicing marks in stone or steel. This is all something that's part of bone taphonomy, Zoo Archaeology, the archaeology of animals. Age, sex, and seasonality, very useful. Biological differences can indicate animal sex. You have spring calves, tooth eruption sequences that tell the age of the animal at the time that they were killed. If we want to assess diet directly from human remains, we can look at some places where humans are exceptionally well preserved, maybe frozen, maybe in a peat bog, where stomach and feces contents are still present. There are examples of human remains in some cases, like the case study Ötzi, the Iceman, in the Alps of Italy and Austria. Ötzi was frozen at high elevation and perfectly preserved. Even the contents of his stomach was preserved, so we could see the last couple of meals by doing autopsy on his digestive tract and retrieving samples. So there's some mummies, obviously, bog bodies. We can also look at tooth abrasions, wear on the teeth of people to get at overall trends in diet. Isotopic studies on hair can be very revealing, or on bone can be extremely revealing of reconstructing dietary shifts of people over the course of their lives. If we're looking at the isotopic methods, we're studying diet over their lifetime, long-term diet and intake. You literally are what you eat to the cellular level, and that is preserved in the organism as long as the preservation factors are there. It's a chemical signature within your body, replicating your diet through time. A couple of different things to point out. For example, you have different types of plants. If you're looking at isotopes, you can look at carbon-3 or carbon-4 plants. Carbon-3 plants like wheat and barley are temperate, and there's a different process for them absorbing and integrating the carbon from the atmosphere than a C4 plant, which is like corn, a tropical plant, even though you may now find corn at elevations such as southern Canada. It is a tropical C4 grass. It originated in the tropics. So these are different isotopic signatures that are preserved in the remains of plants and also are incorporated into the organism that spends a lifetime consuming them. So I thank you for your attention. Here's a couple of credits. This was in part a review of Renfro and Vaughan's Chapter 6, Archaeology Essentials, and I thank you for your time.